Good morning, good afternoon, good evening, hello, bonjour, hola, guten tag, ciao, nin hao, namaste, aloha. I was just in Hawaii. Konnichiwa. We welcome you all to Global Immunotalks for another great round of discussions and insight from our guest speaker today, Keke Fairfax from the University of Utah. Keke was born in Detroit, Michigan, but her maternal family comes from the British Virgin Islands, the island of Tortola. And so that's a place that we often desire to get to visit, but Keke has direct ties to this beautiful area. Keke did her PhD at Yale University, and then she went on to do her postdoc in immunoparasitology at the Trudeau Institute, and also at Washington University. But now at the University of Utah, Keke's lab has been spearheading some amazing work, looking into the elucidation of the immune defenses against the Helminth pathogen, I can always have a hard time saying Schistosoma manasoni. She'll say it much better than we do. We always say schisto for short. But she's been looking at understanding how the humoral response and the B and T cell interactions are generated during this infection to try to lead to better development of immune protection, and has really made some interesting insights into understanding how resident memory B cells are forming and how they can provide local protection, and how the follicular helper cells are involved in regulating this aspect of B cell defense, and is just doing some exciting work in this area, making a foundational new insight into how we think about infection in tissue-specific manners to these Helminth infections. And also more recently has been uncovering intersections with metabolism and how the metabolic regulation of immune cells, particularly macrophages, is affected by the infection, and how that also leads to changes in the pathogenesis of the infection. And more recently, her work is looking at an extremely important area of maternal health, and how infection of the mother in fact impacts the immunity and health of the neonate, and it's just an exciting area. Keke wears a lot of hats. She's very, very busy. She's not only risen to the ranks of associate professor at the University of Utah in no time, but she's also the director of diversity, equity, and inclusion, the co-director of a training grant, co-director of IMPREP. So many, she's very, very involved. And in fact, her work on DEI has been influential and transformative for not only her own local institution, but also for a lot of the colleagues around the country that have been able to interact with Keke in this manner, and to learn from her about mentoring and diversity, and how to improve our climate and our culture in science. She's also received awards already from Regeneron, a Young Investigator Award from the International Cytokine and Interference Society, and the University of Utah has recognized her for Inclusion Excellence Award for research. So hats off to Keke. She's making an impact and being recognized for it. I also just want to say she's an avid Twitter active user, and so you always get to hear what's going on with Keke and how she's in the midst of writing grants or cooking or doing some fun activities. And so it's a great way to get to know you outside of within social media. And so we want to always ask our speakers a question. And so the question that we'd like to ask you, Keke, is what is the trait in your personality that helped you the most in your scientific career?
All right, so that's really my ability to ask very broad questions and think very broadly. And I'll explain what I mean by that. So I do what some people call kitchen sink experiments. And when I was a postdoc, everybody around would label these Keke experiments. And so I might be asking a question about T cells, but I look at B cells, T cells, macrophages, dendritic cells, and I pay very close attention to anything that's changed. And that, really looking at everything is what has led to really the two tracks that have made my lab successful. and that's macrophage metabolism during schistosome infection, and really bringing back the study of B cells and understanding what B cells do in tissue beyond just making antibodies to go systemic. And both of those discoveries were really not the initial outcome measure that the experiment was set up to find and look at when I made the first pivotal observations. And if I hadn't been paying attention, everything would have been missed by me. So that's really something I try to teach trainees, and I think is really important in science.
Yeah, to always look at those odd dots on the flow plots, right?
Yeah.
Dive deep. Yeah, it's great. No, it's beautiful work, and we're really excited to hear you talk today. So thank you so much.
Thank you. Are we ready?
Yeah, go for it.
Okay. Sorry, I have to do this two-step. And I gotta hide my bar. Okay. All right, so today I'm going to talk about macrophages. As Sue said, my lab looks at a lot of different cell types, but I decided not that many people talked about macrophages this year, and so I was going to close out the year with macrophages. And so I'm going to tell a story that's really rooted in thinking about immunology and infections through the lens of pathogenesis, and really both sides of the coin, the host and the pathogen. And so I will give you some background on schistosomiasis, which is not something that most people think about on a daily basis. And so this is a parasitic helminth. We work on Mancini. And the adult worms from Mancini pair, and they live in the portal vein and the mesenteric vasculature. And disease due to infection, all of the pathology is actually not directed against the adult worms, which are relatively immunologically inert in the blood vessels, but it's actually directed against the eggs. And so almost all of the disease that we think of as pathology is really about inflammation around the parasite eggs that are lodged mainly in the liver and then in the small intestines. So the end result in the liver is the development of hepatic fibrosis, and in severe cases, both portal hypertension and pulmonary hypertension. Schistosomiasis is actually the world's leading cause of pulmonary hypertension. And we stereotypically think about schistosomiasis as being a Th2 disease. And that's because it's associated with a marked Th2 response during chronic infection that has eosinophilia, alternative activation of macrophages, and in mouse models, the production of IgG1 and IgE from the antibody side, and in humans, IgG1, G4, and E. And so this disease really happens because the eggs would like to go down, break out across the lumen of the gut, and be excreted in the feces. But unfortunately, in all mammals, blood flows up. And so that's why you end up with all of these eggs lodged in the liver and the vast majority of the pathology that a host experiences. And so I said that, we think about schistosomiasis as a Th2 skewed disease. And that is true to some extent. And it's really been for decades one of the most well-studied diseases for understanding immunomodulation. And that's because there are distinct phases in the immune response to infection. So early on after infection, egg laying begins at about 5 weeks post-infection. You have a large immune response that is a mixed response, both Th1 and Th2. And you get really large granulomas made against those eggs that are deposited. You have the generation of both type 1 and type 2 effector T cells. And you have this really strong immune response. But in order for the host to survive, you actually have to downregulate this immune response and sort of stabilize both your T cell levels and your B cell levels, as well as the actual myeloid response around the eggs in terms of granuloma size. If you fail to do that, the host gets severely ill and usually dies because they've lost too much liver function. And so when we think about type 2 immunity in schistosomiasis, most people are really thinking about this window of chronic infection. But there was a lot when I started working on schistosomiasis that we really didn't understand about this modulation, and it was really viewed through a T cell lens. And so some really important work that ended up being informative for everything I do in my lab now, both the B cell projects and the T cell projects, was work I started as a postdoc to really understand this transition to chronic disease and what were the key drivers of chronicity and the host down modulating their immune response. And what we discovered is that it's actually driven by tissue resident B cells through a macrophage effector function. And so before you get chronic infection, so this is nearing the end of the acute stage at 10 weeks post-infection, these are liver confocal sections. You can see an egg here with its stereotypical spine, and there is antibody around the egg, but then you have this core large granuloma of F480 positive macrophages. And you can see this repeated here. Here you're just at the top of the granuloma. Once you get into 16 weeks post-infection, you can now find that these granulomas are more compact, as you can see here compared to here. And all of these macrophages are ligated on their FC receptors with immune complexes, right? So they're all IgG1 positive. And we had known for a while that IL-10 was really important in infection. So this project was actually originally set up to look at the role of IL-10 in T cell modulation. And it turns out T cells aren't modulated if you block IL-10 signaling during chronic infection. But what happens is you dysregulate this B cell macrophage axis that controls the granulomatous response. And you end up with what are called coalescing granulomas, that now the macrophages do not have their immune complexes, their FC receptors ligated by immune complexes, and we ended up with actually pulmonary hypertension in these mice that were blocked during chronic response of IL-10 signaling. So this identified to us that it's really a key interaction between B cells and macrophages in the liver that are protective on the host side from developing severe pathology. And this really set us up to look at and really understand what this macrophage response is really about, right? What's the difference between these macrophages at 10 weeks versus these now that have their immune complexes ligated and now, normally, and now make much more compact granulomas and are not pathologic to the host, right? And when this process doesn't happen, you get severe host pathology and, most of these mice die. And so this really started us on this task to understand what do macrophages look like during schistosomiasis from a transcriptional standpoint. And this is work that we published in 2018 and started as part of my work in the IMGen consortium, where we set out to look at the heterogeneity of liver macrophages. And we did that both at steady state and in the context of schistoinfection, because I had this nagging question of what does this immune complex ligation do to the macrophage, and why does it now become less pathologic to the host? And so when we were looking at the transcript data from sorted both Kupffer cells and perivascular macrophages, I really expected to see a profile of inflammatory mediators, but instead what we found was a dramatic remodeling of metabolism in these cells. So both the biosynthesis of amino acid pathways as well as the glucagon signaling pathways were dramatically altered in both perivascular and Kupffer macrophages sorted at 12 to 14 weeks post-infection. And we also had a dramatic remodeling of cholesterol synthesis pathways. And so this really started to suggest to us that what we thought was a story about inflammation might at its heart be a story about metabolism instead. And all of this work was done in the time frame where it's really pivotal work about M1 versus M2 macrophages, really hinging on cell intrinsic metabolic drivers, of inflammatory mediators. So this was extremely interesting to us. And so we started moving into a model of metabolic disease that's very commonly used. And we use the ApoE mouse model system. Now, these mice are really great because in most animal facilities on high-fat diet, They will get, of course, atherosclerosis, which is what they were originally generated to study, but they also become obese and they get type 2 diabetes. They become insulin resistant. And so we started doing experiments with schistosomiasis in this ApoE knockout background. And what we found, so the white open circles are chow uninfected control mice. The black triangles are high-fat diet uninfected mice, right? So these are the ones with severe metabolic disease. And then we have high-fat diet mice that are infected, and then the chow infected controls. And what we surprisingly found is that both with and without metabolic disease, if you look at this chow infected, blood glucose levels in response to a glucose tolerance test are dramatically modulated in the context of schistosome infection. And so the chow uninfected have the lowest both fasting glucose level and the lowest response curve to a glucose tolerance test. But then you have significant protection driven by infection here of glucose intolerance. And we can quantify this with area under the curve of glucose. And you see it's significantly lower in the high-fat diet infecteds. And on normal chow, an infected mouse has a much better glucose tolerance than an uninfected mouse. And so we really started to approach this through the context of macrophage metabolism. And why do we care about this? Well, it's been known for about 15, 20 years that globally, Helminth infections inversely correlate with metabolic disease. This is true for schistosomiasis. This is true for ascaris. This is true for trichuris. This is also true for filarial helminths. And so this really seems to be a phenomenon of parasitic Helminth infection. And so why should this be interesting to us in high income countries where we don't have schistosomiasis or any other parasitic Helminth really endemic anymore? And there we started thinking about it through the lens of what are the major disease drivers in high income countries? And it's really metabolic disease. So more than 65% of diabetic patients die from cardiovascular complications. Insulin resistance and obesities are two of the most important risk factors for the development of coronary artery disease in diabetics. So there's really a critical need to develop innovative approaches to reducing the pathology of metabolic diseases. So this is absolutely true in high income countries, but this is actually unfortunately becoming more and more true in lower middle income countries as well, where we have increasing rates of metabolic disease in these countries as they shift to a much more Western diet. And so all of the rest of the data I'm going to talk about today, which is both published and unpublished work, is using this ApoE mouse model. And we're doing almost all experiments at 10 to 12 weeks post S. mansoni infection. All mice are pre-started on diet approximately 10 days before they're infected or mock infected. And this is the standard Western high-fat, high-cholesterol diet that many people use for ApoEs and LDLR knockout mice. And we really take an integrated omics approach in all of these studies. So we're looking in many, many, many compartments. We're looking in the plasma, we're looking in the liver, we're looking in the bone marrow, and we're looking in blood for BMCs. And we do a lot of different measures. We heavily rely on both transcriptomics, metabolomics, and lipidomics. And we've tried really over the last 4 years to take an integrated analysis approach that considers the transcriptome in light of the metabolome and the lipidome, and vice versa. All right, so I'm going to start with the most striking piece of data that we got approximately 6 years ago. And that's we had these mice, ApoE knockout mice on high-fat diet. These are all males, either infected with S. mansoni or mock infected. And we just took out their bone marrow at 10 weeks post-infection, cultured it, no antigens present in the culture, just cultured with MCSF to make bone marrow-derived macrophages, and then used seahorse assays to measure oxygen consumption and extracellular acidification to start to look at the real-time metabolism of these macrophages. And what was really surprising is that both basal oxygen consumption here and quantified here, as well as spare respiratory capacity quantified here, when these cells come from an infected male mouse, is dramatically higher, right? So this is a quantitation of basal OCAR, and then this is spare respiratory capacity. There is no change in extracellular acidification. So there isn't really a lot of lactic acid lactate being made from these cells, which is usually an end product of glycolysis. And so I told you, over the last 8 years, there's been a lot of looking at macrophage metabolism in the context of type 1 polarization versus type 2 polarization. And it's previously been shown in M2 macrophages that they undergo cell-intrinsic lysosomal lipolysis, and it drops their lipid droplets way down. And that's not happening in these cells. So we have no change in bulk oil red O staining as measuring lipid droplet size. But we do have a dramatic increase in mitotracker MFI, which can be linked to either increased mitochondrial function at the individual mitochondrial level or mitochondrial biogenesis. And so this is just to demonstrate to you that these really are unpolarized cells. So this is CD301 by 206, which we look at normally double positives are M2 macrophages. These are not M2 macrophages. They're really all M0 unpolarized macrophages. But we used bulk RNA sequencing of these macrophages to start to ask what is altered at the transcriptional level in these cells that might underlie this increased oxygen consumption at a basal level. And we found a lot of genes changed, actually, and some of them very surprising. Some of them are typically associated with interferon responses, so GBP6. This is downstream of interferon responses. There are genes like monoacylglycerol lipase, MGLL, which can generate free palmitate from monoacylglycerol, as well as free fatty acid receptor 2, are all significantly upregulated. And then we have other genes that work from our lab and others have shown to be associated with type 2 development of macrophages and in the prostaglandin pathway like HPGD. But these genes were really surprising here, particularly these ones downstream of interferon, coupled with this monoacylglycerol lipase. And so when we looked at pathway analysis, we found a large number of genes downstream of interferon gamma that are upregulated in these cells. Again, these are not stimulated with either LPS or SEA. This is just MCSF culture. But we could never find an increase in actually interferon gamma transcripts or interferon gamma protein. But there is a very strong inflammasome signaling pathway as well as chemokines. And so this started to suggest to us that there's a very dramatic reprogramming of both the inflammatory potential of these cells as well as the metabolic potential of these cells. So I told you one of the genes that really stood out to us was MGLL. And so we actually wanted to test the role of MGLL in this increase in both basal oxygen consumption and max respiration or spare respiratory capacity, depending on how you do the calculation. And so we used an inhibitor of MGLL, and that's shown here. And so these are control cells from uninfected males without the inhibitor, and with the inhibitor, there's no change to either basal oxygen consumption or max respiration. When we take cells from infected males and culture them without the inhibitor, we have the same increase in both basal oxygen consumption and max respiration I showed you in the previous experiments. But when we add the MGLL inhibitor, we bring this down to the exact level statistically of both basal oxygen consumption and max respiration we see in control cells. So this tells us that MGLL is really necessary for this phenotype of increased basal oxygen consumption and metabolism. And MGLL, as I told you before, is a monoisoglycerol lipase. It generates a free fat from monoisoglycerol as you go through tri, di, and monoisoglycerol cascade. And so this really suggested to us that these cells have dramatically different needs for lipids compared to control cells. And so we did untargeted lipidomics. And then we looked at the types of lipids and fats that were altered. And we found a dramatic decrease in cholesterol esters in these cells generated from bone marrow from infected males, with a very surprising increase in all chain length of free fatty acids, short chain, long chain, and polyunsaturated free fatty acids. And again, I want to, put this back in context with the literature, where this is actually the exact opposite of what has been published by multiple labs for M2 polarization, where they actually burn fat and they do not accumulate either free fatty acids or cholesterol esters in their lipid droplets. So this is dramatically different. And I'm not going to show it to you, but we did show in this paper from 2021 in January in PLOS Pathogens that this is actually the opposite phenotype of if you administer IL-4 immune complexes to a mouse for the same amount of time, right? So this is very distinct from what IL-4 or alternative activation does to the potential of a bone marrow macrophage precursor. And so we wanted to directly assay the ability of these cells to use fats. And so we use palmitate conjugated to BSA as a carrier to get it across the membrane for these experiments. And again, these are seahorse assays. So here, the only carbon source available is this palmitate conjugated to BSA. There's no glucose in these cultures or glutamine. And so when you have the cells from infected males and their only carbon source is palmitate conjugated to BSA, you see this dramatic increase in both basal oxygen consumption and spare respiratory capacity, similar to what we saw with a normal seahorse assay where glucose is the carbon source, right? So this suggests that these cells are completely capable of using palmitate as their sole source for carbon. That is not the case for cells from uninfected males. They have very low levels of basal oxygen consumption and a minimal amount of spare respiratory capacity when palmitate is the sole carbon source. When you add this inhibitor, CPT1, which inhibits when you add etomoxor, an inhibitor of CPT1, you dramatically reduce both this basal oxygen consumption and the spare respiratory capacity in the cells from infected males, and this is here in the light blue, with very minimal effect on the cells from uninfected males. Almost nothing to the basal oxygen consumption and a little bit to the spare respiratory capacity and max respiration. So we can quantify this CPT1 dependent, both basal oxygen consumption and spare respiratory capacity by subtracting these out. And you can see that the cells from infected males have a significant amount of their basal oxygen consumption dependent on CPT1. And same thing with their spare respiratory capacity as compared to the cells from uninfected males. So again, taken with the MGLL inhibitor, this really suggests that these cells are set up to generate palmitate from their own stores of monoacylglycerol and then use that for their oxygen consumption needs. And when you inhibit that, you dramatically alter both their basal oxygen consumption and their stressed spare respiratory capacity of the mitochondria. And so the question then we want to answer, right, is that I told you that these are cells that we generate macrophages in culture in a standard 6 to 9 day BMDM culture with MCSF, which was suggesting to us that this is metabolic reprogramming that's happening, long before it becomes a macrophage, right? If we can see this in a dish without antigen present. But we wanted to directly ask, how long can this phenotype last in the absence of antigen? And then whether or not these cells can actually directly modulate systemic metabolism in a host. And so we used bone marrow chimeras. And so we pre-treated recipient ApoE knockout males on high-fat diet for 5 weeks to get started metabolic disease. And then we've done this 2 separate ways. We've either irradiated them or we've used busulfan to deplete the myeloid progenitor compartment. And we use human-grade busulfan that we get from the bone marrow transplant clinic. Works very well in mice. And we get the same data with either way we do this experiment. And so after either 5 days of busulfan treatment or lethal irradiation, and then we transfer in donor bone marrow from either uninfected males that have been on high-fat diet for 10 weeks, or donor bone marrow from infected males that have been on high-fat diet for 10 weeks. And then we park it there for 10 weeks, and these recipients are kept on high-fat diet, and we're looking at systemic metabolism, and then we're also going to take the bone marrow back out and look at it. So if we look at systemic metabolism at 10 weeks post-transfer, we do see a protection in the face of a glucose tolerance test. We have reduced glucose area under the curve. The body weight also trends lower. The p-value of the body weight, I believe, is like 0.056. It's very close. And then when we, more importantly, take this bone marrow back out and then culture macrophages with MCSF, we see the same phenotype as increased basal oxygen consumption and spare respiratory capacity, and this is quantified here. And so this bone marrow, after 10 weeks of parking in the absence of schistosome antigen, only the high-fat diet, is able to retain this profile of increased basal oxygen consumption and spare respiratory capacity, and most importantly, increased mitotracker MFI. And again, this mitotracker that we use measures mitochondrial mass. And so this suggests that this reprogramming is really long-lived, right? And so when we think about what is present in the bone marrow that can generate a macrophage when you put it into culture with MCSF, there are of course monocytes, and the half-life of monocytes is significantly shorter than the 10 weeks of this experiment. And then there are both short-term progenitors like GMPs, as well as hematopoietic stem cells, all capable of differentiating into cells that can make a macrophage eventually. And so we're very interested in what progenitor level this reprogramming is happening at. And I'm going to get to that in a couple of slides. But first, I want to go back to, I framed this as really understanding pathogenesis, right? So what does this do for the host? What does this do for the parasite? And looking through that lens, we started to ask questions about, well, from the perspective not of metabolism, but perspective of the host response in terms of a granuloma size and reducing their own pathology, what would happen if you altered the macrophage response to schistosome egg antigens, right? So I showed you earlier the pictures of granulomas at 10 weeks, which is at the end of the acute stage, and at 16 weeks when you're solidly in chronic infection. And I hope you appreciate that those granulomas look very different from each other, right? And so granulomas are made up of macrophages as one of the major cell types, in addition to eosinophils and neutrophils. And those macrophages we've published and other labs have published, once you're past about 6 weeks post-infection, so the very earliest time points of the immune response, all of those macrophages are actually coming from blood monocytes, not from proliferation of Kupffer cells or perivascular cells. And those blood monocytes, of course, then turn into new perivascular macrophages and new Kupffer cells. But they're, a very different cell type than what was present in the liver before infection. And so we asked the question of, if we have a macrophage that has never seen schistosome egg antigens, how does it behave, right? And that's going to model an early granuloma, right? Very early during infection. Versus how does it behave if it's generated in the context of having seen schistosome egg antigens back when it was a progenitor in the bone marrow for a long period of time, right? And so we do this by taking, again, bone marrow from either control males or infected males on this side. And then we stimulate it after generating a mature macrophage in a 6 to 9 day culture. We then stimulate it for 24 hours with SEA. And this is just showing you pathway analysis of upstream regulators based on the differentially expressed genes. And so when a macrophage sees SEA for the first time, there's a really strong pathway actually of interferon gamma, IL-10, STAT3, TLR4. There is a pathway of IL-4, but it's pretty low down. And interferon gamma and STAT3 seem to be extremely important here. But if instead this macrophage was generated from bone marrow that came from an infected mouse, right? And I just showed you that that bone marrow is absolutely reprogrammed, both its transcriptome as well as its metabolic potential. And then it sees SEA, it has a dramatically different response. Now this dominant response is actually interferon gamma downstream pathways. And TLR4 comes up, and now STAT1 as opposed to STAT3. And IL-10 is of course down here, but it's much fewer genes than interferon gamma is. And so this started to suggest to us that, you know, this regulation of metabolism is really like an aside, a bonus, right? What maybe the worm is really trying to do is protect the host, right? And so if you modulate the progenitors in the bone marrow so that when you have new monocytes that can then go to the liver, go to the intestines, and enter into this granulomatous reaction, and you've dramatically reprogrammed both the inflammatory mediators that they're going to make, as well as their metabolism, which underlies lifespan and a lot of other things, maybe you can protect the host from severe pathology by doing this. And so this is something we're extremely interested in, and the rest of the data I'll show you is how we're starting to get at this from a metabolic standpoint in terms of what the metabolism might be doing for this inflammatory immune response here. So I'm going to give you the conclusion so far, and then everything else I'm going to show you is unpublished, and that's previous slide was unpublished. And so we really got to an understanding that bone marrow-derived macrophages from male S. mansoni mice have increased basal oxygen consumption and spare respiratory capacity. Myeloid reprogramming in male mice leads to a dramatic increase in mitochondrial fatty acid oxidation. And this increase in fatty acid oxidation is supported by both importation of free fatty acids, and that's that upregulation of free fatty acid receptor 2, and MGLL-driven lipolysis, which is a very unique form of lipolysis in terms of myeloid cells. I didn't show you this data, but it's in that 2021 paper. There is an alteration in the response to LPS stimulation, both at the transcriptional level as well as the secretion of inflammatory mediators. And most importantly, this reprogramming is long-lived in the absence of antigen and is transferable to an uninfected host. And so everything I showed you in the first set of slides was all in male mice. And that's because, you know, I'm equally as guilty of it, about 6 years ago. Like many people that do schistosomiasis, we typically have only used male mice. And this is true in a lot of other infections as well. And when you looked at the epidemiological data, most of the epidemiological studies that have really drawn this link to Helminth infection and regulation of metabolic disease and metabolic mediators like BMI are really underpowered for females. They're usually mostly male studies. And if they have females, they're statistically underpowered to have identified a sex effect. And so we started infecting female ApoE knockout mice, and we were very shocked that female ApoE knockout mice are neither protected from obesity nor glucose intolerance. This is not statistically reduced. This is actually highly variable. I'm showing you the worst glucose area under the curve that we have in terms of there's slight protection here, but it's not statistically significant. Most of the time, these lines are pretty much identical. And when we look at very important mediators of cardiovascular disease, which are serum triglycerides and diglycerides, we see that male mice, when they're infected with S. mansoni, have a reduction of both triglycerides and diglycerides in the serum, but females actually have a significant increase in triglycerides when they're infected with S. mansoni, and no change to diglycerides. And so this really suggested to us the opposite pattern of metabolic modulation in females compared to male mice. And so, our previous data had really suggested that there was reprogramming of progenitors going on based on both our bone marrow cultures and our chimeric mice. And so we started to think about hematopoiesis. And so this is just a reminder of bone marrow hematopoiesis. They're hematopoietic stem cells that are pluripotent and can self-renew. And then you differentiate into a series of more and more restricted progenitors. And the myeloid line is classically thought to come from MPP3s, which make the common myeloid progenitor that can then make both the monocyte dendritic cell progenitor and the granulocyte macrophage progenitor here. In emergency myelopoiesis, as it's been studied, it is possible for MPP4s, which are also called LMPPs, which normally seed the lymphoid progenitor line. In emergency myelopoiesis, so these are conditions of extreme inflammation in the bone marrow, there's a FLT3 ligand-dependent way for LMPPs to form either straight GMPs here, the granulocyte macrophage progenitor, or to first make a common myeloid progenitor and then differentiate into a GMP. And this is well studied in leukemia, but happens in a lot of contexts. And so we asked, at what stage are we seeing an alteration to progenitors? And so the first stage that we found a significant difference were in GMPs. So we have an increase in Ki67 positive GMPs that you can see here in these male mice. This does not occur in female mice driven by infection. And then if we look at LMPPs, which I told you in emergency myelopoiesis can make GMPs, we see a significant increase in LMPPs in both frequency and absolute number. The absolute number is even more dramatic, only in male mice. And so we really think that LMPPs are forming GMPs and then eventually macrophages in culture and in the whole body mouse in the context of schistosome infection in male mice. And that emergency myelopoiesis pathway is not happening in females. And so we, of course, wanted to know what is the transcriptional profile that underlies this dramatic difference between males and females. And so we did RNA sequencing on males versus females, bone marrow derived macrophages, and we see that regardless of sex, so this is looking at just genes that are upregulated in males, and this is upregulated both in the uninfected males versus females and the infected males versus females. We see there's actually a large number of genes that are sex specific in the context of macrophages generated from bone marrow. And the majority of these are actually metabolic genes. And then when we look at differential regulation, these are genes that are upregulated in males in response to schistosome infection compared to females. We actually see, again, a fairly large number, considering that this is the same parasitic infection. And there's 66 genes. The vast majority of them are metabolism genes. Many are not assigned at the KEGG pathway level to metabolism, but when we look at their functions, they're really metabolic disease genes. And then 10 that are traditionally assigned to KEGG metabolism pathways. Very interestingly, one of those is MGLL, which we identified as having an important role in both basal oxygen consumption and spare respiratory capacity in cells from infected males, as well as hexokinase 3, which can induce mitochondrial biogenesis, and DO2, which has a strong role in metabolic disease in humans and in mouse models. And so it really indicated to us that there is a huge dependence on sex for this reprogramming that we see in macrophages. So does this bear out in the lipidome, right? So I told you one of the canonical features of these macrophages derived from infected males was this decrease in cholesterol esters. So again, these are separate cohorts of mice where we now have males and females together. And we still see this dramatic decrease in males. And then interestingly, a dramatic increase in cholesterol esters in infected females compared to uninfected females. And I do want to acknowledge here that the uninfected females are dramatically different from the uninfected males, right? So this is truly rooted in biological sex. This relative abundance of free fatty acids, as I told you, it's dramatically higher in infected males, in bone marrow derived macrophages from infected males compared to uninfected males, and this does not happen in females. And again, at the basal level, even before infection, there is a difference between males and females. Females have more free fatty acids in their macrophages. And so the final aspect we looked at here was this ability to use palmitate as a carbon source. And female cells, regardless of whether they come from an uninfected animal or an infected female, are really poor at using palmitate as a carbon source. This is the exact same assay as we did in males. This is palmitate conjugated to BSA, and it's the sole carbon source. And so they're really poor at using palmitate as a carbon source with very little basal oxygen consumption and very little spare respiratory capacity. And again, this is the opposite of what we see in males, right? So all roads are leading to the real-time metabolism and the ability to use various carbon sources of male and female cells being very different, and then being differentially regulated by infection. So we have a working model where in males, infection drives reprogramming of myeloid progenitors that leads to macrophages that have increased DO2 and hexokinase 3, PFKB3, which I didn't talk about much, but is really important in glycolysis. And we do actually have an upregulation in glycolysis in male mice. And if you read the 2021 paper, we really believe that that upregulation in glycolysis is really about generating palmitate as lipid carbon source, and an increase in MGLL. We see essentially the opposite thing in females. We have a decrease in DO2, a decrease in hexokinase 3, a decrease in PFKB3, as well as MGLL. And then you end up at the cellular level with decreased cholesterol esters in males, increased free fatty acids, increased mitochondria function, and likely more mitochondria. And all of the carbon sources are really being used to make intermediates for the TCA cycle and fuel fatty acid oxidation in the mitochondria. And this is correlated with protection from high-fat diet-induced weight gain and glucose intolerance. And so you have to think about what's different between males and females, right? What could be responsible for this differential response to schistosoma mannsoni antigens? Of course, there's chromosomes that are different. Females have 2X chromosomes, males have a Y chromosome, and of course, sex hormones. And so we started to address the hormonal level by performing ovarectomies and castration before we infect the mice. And so here I'm showing you body weight and then glucose area under the curve. In black, we have uninfected females on high-fat diet, either intact or overectomized. In blue, we have infected females on high-fat diet, either intact or overectomized. As we would expect, and this also happens with humans when women undergo menopause, there is weight gain associated with overectomy. And infection not only protects from this ovarectomy-induced weight gain, but actually brings the weight to below what the intact infected female is. More importantly, from our standpoint of being interested in macrophage and whole body metabolism, is that the glucose area under the curve, as I showed you before, is statistically identical in intact females, whether they're uninfected or infected. But then when you overrectomize this female, you significantly lower the glucose area under the curve in response to an IP glucose tolerance test. And this lowering is actually statistically significantly lower than the intact mouse as well, as indicated here. So this is truly bringing metabolic protection to the females via ovarectomy before they're infected. What happens with males? Well, actually castration doesn't do much from a metabolic standpoint. And so there is, of course, a loss of body weight when males are castrated. As you would expect, the loss of testosterone generally decreases muscle mass. So this is true for both uninfected animals as well as for infected animals. They have lower overall body weight, and most of this is a loss of muscle mass. But when we look at their glucose area under the curve in response to an IP glucose tolerance test, we see that both the intact and the castrated infected males are statistically identical to each other, and they both have the same degree of reduction of glucose area under the curve compared to the intact animals. So testosterone is not needed in males in order to have protection from glucose intolerance induced by high-fat diet. Whereas in females, ovarectomy allows them now to have protection from glucose intolerance. And so what led us here was really about cellular metabolism, right? And so we believe the most important outcome measures are really about this macrophage metabolism, as it underlies both inflammatory function as well as can directly affect the metabolic parameters that we look at a systemic level in terms of body weight and systemic glucose tolerance. And so I told you a few slides ago that the hallmark really of males that cells that come from males infected with S. mansoni is this fuel flexibility, right? They can use glucose, they can use palmitate, they can use glutamine, all feed fatty acid oxidation. And that was not the case for females, right? And so if we zero in on just the females, either overrectomized and uninfected or overrectomized and infected, we see that infection normally in an intact female, as I showed you before, doesn't increase glucose respiration, palmitate-induced respiration, or glutamine-based respiration. And these are all single carbon source assays. So these are seahorse assays where either glucose is the sole carbon source, so there's no glutamine. We use dialyzed serum so that there is no fat, or palmitate is the sole carbon source, or glutamine is the sole carbon source. And so what should be most striking from here is that overrectomized infected females have significant increase in maximal respiration, which is that first peak of the spare respiratory capacity curve, when the carbon source is either glucose, palmitate, or glutamine, right? And they're all relatively equal in terms of respiration rates. And so this really suggested to us that just removing hormones from these females now allowed their progenitors to be reprogrammed to have macrophages, when they're generated, have dramatically altered metabolism. And so this is one of my last data slides. And so we said that this was really about lipids, right? That we believed that all of the metabolic carbon source outputs were being used to fuel fatty acid oxidation. And so this is essentially pathway analysis for lipidomics data. This is really, really cool software. So when we look at the lipidomic data from infected males versus infected females, there's a strong signature of free fatty acids 22.5 going into 22. And so these green bold lines are all statistically significantly increased. And so this is the signature of male lipids from the macrophages, and it's very different from females. Now, when we overectomize females and they're infected, and we can compare that to uninfected overectomized females, now these infected overectomized females again have this strong signature of going into free fatty acid 22. This isn't identical to males, but it really starts to phenocopy the male pattern and suggests to us we've dramatically reprogrammed lipid metabolism in these female cells just by overectomizing them before they're infected with schistosomiasis. And I wouldn't expect it to be identical to males because obviously there's still extreme genetic differences between males and females. But it does indicate that the hormones that are normally present in the females are dramatically reducing the ability of schistosome antigens and the milieu of schistosome infection to reprogram progenitors. And removing this one break is able to get you, 75% of the way to looking like a male. And so our conclusions thus far from all of the data I've shown you today really focuses on this novel finding that S. mansoni infection differentially regulates both myelopoiesis and lipid and cholesterol metabolism in males versus females in response to diet. We know that the male reprogramming begins at least at the LMPP level. Ovarectomy alone is the only intervention that's needed in order to allow females to phenocopy the myeloid metabolic phenotype of males and be similarly reprogrammed. And importantly, in males, this isn't dependent on testosterone, right? You don't have to have the normal male complement of hormones in order to be reprogrammed in response to schistosome infection, because castration does not alter this. And most importantly, once you've overectomized a female and then infected it with schistosomes, they now have this fuel flexibility and this ability to burn lipids that was really only present in the males before. So again, one simple intervention of removing female hormones is able to dramatically reprogram myeloid metabolism in response to schistosome infection, which is extremely novel. And I will end there and acknowledge all of the really important people that did this work. So Diana was a graduate student who graduated in 2019, and she was the first author of the 2021 paper. Lisa Gibbs is a current graduate student, and she will be the first author of the paper that we will be submitting soon, that is all of the unpublished work I've shown you, along with a postdoc, Juan Marcos Oviedo. And I have a talented honors undergraduate student who's also been working on myeloid cells here. And very importantly, you can't do any of this without my lipid and metabolomic gurus who are very important contributors to this project, James Cox and Alan Maschek, and my dear friend and co-conspirator in all my macrophage work, Eyal Emil, who has a lab at the University of Vermont. And all the work I've showed you has been funded by the NIH and the American Heart Association. This is Diana, and this is Lisa, and that's Juan. And I will stop there.
Great. Thank you so much, Keke. It's just fascinating how different it is with the, and what nuclear hormone receptors likely will be involved in driving that shift. That's going to be, and how it will apply to other infections. A lot of people aren't looking at these gender differences between during infection. So I think this is really fascinating work. Thank you so much for that. Everybody who would like to ask Keke questions, she has her own Twitter account. So please, you go to @LabFairfax and add your questions, and she'll reply to them in the next few days. Of course, stay tuned to Keke's next adventures. She's, like I said, she's an active Twitter user. And I think at this point, Carla and Alina would like to say something.
Yes, thank you. Thank you, Sue, and for hosting Keke, and thank you, Keke, for a wonderful, wonderful talk and sharing such important science. So, yeah, I have a big announcement to make, and then I will pass it to Carla. But before I do that, and announce the new organizers for 2023, I would like to first emphasize that we are super grateful for all the help that Matteo Iannacone and Florent Ginoux have done for Global IMMUNOTALKs in the past 2 years. They are stepping down as organizers of Global IMMUNOTALKs in 2023, but they will continue supporting Global IMMUNOTALKs as members of the Scientific Advisory Board. And so we are super grateful for that as well to them, both for their continued support. And as always, in the last Global IMMUNOTALK session of the year, we want to announce the new organizers for the year 2023. And this is Dietmar Sen from Germany and Kazuyo Moro from Japan. And so we continue being global, and we are super, super grateful to the 2023 organizers, both Dietmar and Kazuyo, and also our amazing organizers, Kate, Burkhard, and Laura, who has been helping us already in 2022 and have done a terrific job. So we are looking forward to a great program in 2023, and Carla will say more about that now. Thank you.
Thank you, Elena. Such a pleasure to connect. And indeed, we are towards the end of the year. And as it has become almost a tradition by now, it's a pleasure to present to you the upcoming schedule for the first semester of 2023. Our organizers have done an incredible job to line up fabulous list of global immunospeakers. So we just say a little bit goodbye by now, but we look forward to joining everybody again on January 18th with James Chen, which will start our new Global IMMUNOTALK series for the year 2023. So thanks, everybody. Happy holidays. And thanks, Keke, for a fantastic talk.
Happy New Year.
Happy New Year.
Happy New Year.
I think